The second talk today is by Zorana Zeravcic. She's an associate professor at ESPCI Paris, and it's going to be in the theme of biology inspiring artificial chemistry, which may even hold clues to how life would work on other planets. Zorana?
Does it work? Okay, perfect. Yeah, so now for something completely different. So we are going to talk about artificial chemistries. So what you see here in this image is a zoom into the Orion Nebula, which was made by the Herschel Space Telescope. So it is in the infrared. And so by analyzing the spectrum, what they found, astrophysicists and astronomers, they found ionized carbon atoms in its center. And in one of his TV episodes about cosmos, Carl Sagan said that we are all made of star stuff. And what he actually means by this is just an observation. It sums the fact that carbon and nitrogen and oxygen and some other heavy atoms were all created in previous generations of stars, some 4.5 billion years ago. Now, life as we know it is exists in a multitude of forms, but when broken down to the most basic elements, it is the same star stuff that we just mentioned. So we have carbon bonded to hydrogen, oxygen, nitrogen, and elements like sulfur and phosphorus. Now, nucleic acids like RNA and DNA contain all these basic elements, basic building blocks. If we zoom in a little bit more, some amino acids and proteins are built by elements up to sulfur, others by elements up to nitrogen. Carbohydrates and lipids are mostly made out of hydrogen, oxygen, and carbon. And then the hydrocarbons are made out of the first two elements. Now, all these building blocks give us these beautiful life forms like this tree or this little echidna or the leafy sea dragon or the dragonflies. So life forms that are very, very broad and diverse, but made just of these very few building blocks. And so in the words of Darwin, it is interesting to contemplate a tangled bank clothed with many plants and many kinds, with birds singing on the bushes, with various insects flitting about, and with worms crawling through the damp earth. And to reflect that all these elaborately constructed forms have been produced by laws acting around us. So really a central question for scientists from different fields, from biology, physics, chemistry, engineering, that are interested in the life around us, so someone like me, you're interested in what are the rules and laws behind what we see in nature. So we want to understand really the physics of life. But now the life as we know it is just basically one possibility, one instance that we know. And really to have a theory of what is actually, it is necessary to understand what are all the possibilities. And these are the thoughts that spurred the field of artificial life, which is basically a synthetic approach to biology, exploring not only life as we know it, but also life as it could be. Now, what has guided my research into the what we call an unknown over the past years is inspired by nature, but not limited by what we see in nature. And so by limited, what I mean is that I'm not considering only the building blocks of life as we know it and reaction that we know. So we lift those constraints and we ask the questions, what are the necessary ingredients we need to achieve a desired lifelike function or property, but without the limitations. So what we assume is that we have available any type of a building block with any type of interaction and reaction possible, but they all have to follow laws of physics. So in other words, we're interested in necessary ingredients to achieve desired functions, but in physically realizable systems. We're also interested in how we can control different levels of organization of artificial matter in order to mimic life. Now, we do all this with the goal of finding efficient and robust design rules at different scales. Why we want to have things being efficient, this is sort of intuitive, but robustness, really, this is the property of life. And everything that I've said basically relies on the fact that we need to have these artificial building blocks, many different types. And because of that, for many decades, research in artificial life has either been limited to just biological systems that we know or computer programs and simulations. But now in the last 10 years or so, this changed dramatically. And so now we can do many more things. And this is what I want to show you. So what properties of building blocks we can control and we actually need to control? So we need to control how isotropic they are or anisotropic, right? We want to control the valence and how they interact with things. And so one of the main ingredients that allows us to make these artificial building blocks in artificial chemistries is DNA. Now, DNA strands act as a specific glue. If you take a single strand of some length, you know immediately what is the complementary strand that will make the double helix together with the first strand. And so for perfect binding, it has to be complementary strand. Now, if you have a strand of length 10, you have over 1 million different sequences that you can make, or basically 1 million different glues that you can make, artificial building blocks. So here I'm just showing a few examples in the experiments that people have done. So the DNA can be used as a specific glue. So basically what researchers do, they take these DNA strands and they put them on surfaces of nano and micron size particle, like what is shown here in the first 3 images. So here the different color means that the particle have different types of interactions, different types of DNA on itself, and then they can bind only if they have also the complementary strands. Now, DNA can be used as a building block, and this is now shown in these next few images, where by in a combination of long and short strands, people can assemble basically any type of any building block of any type or any shape that you can imagine, by just carefully designing the sequence. Now, this is not the only way to make artificial building blocks. You can also play with just purely shape. But this is, I put here a few images, but they will not be needed for the rest of the talk. Now, another important thing about DNA interaction is that they can be tuned and turned to be on and off by changing the temperature. And so at which temperature basically this happened really depends on how you design the sequence. So now, all this, what I've just said, makes my life as a theorist easy, because I can just assume that indeed we can have any kind of a building block that we want. We can design any kind of interactions that we want. And so the model system that I work with is just based on spherical particles coated with DNA. So they're usually microns, like we imagine micron-sized particle, and then the DNA is on the nanoscale. So basically we are talking about very short-range interactions. Now, another thing that is quite important for what I will tell you, and that doesn't exist in all the systems, is something that we call species valence. And so with the particles which are either emulsion droplets or spherical particles coated with some lipids on the surface, when you graft the DNA strands, they can actually be mobile on the surface. And it was shown that when 2 droplets come together, these DNA strands basically are mobilized and they move to make a bond. And by controlling the density, particle valence can be controlled. And so in this particular example, what you can see is a yellow particle that has enough DNA that can bind 2 red particles, and red particles have enough DNA that can bind only 1 yellow particle. And so if we make a solution that contains a mixture of these particles, what we will end up having are monomers, dimers, and trimers that are floppy. Okay, so now that we have all these building blocks, the next step is to figure out what kind of nature processes we would like to study and explore beyond what is observed, and to find the laws and rules that govern them. And so within this bio-inspired design, one wants to understand the process of self-assembly, or the ability to spontaneously make complex structures from simpler parts with high fidelity. Then one wants to understand the process of self-replication and error correction. The first being the ability to make copies of itself, and the second, to correct errors made along the way. And finally, if we could be able to mimic metabolic processes, which basically are to construct self-sustained catalytic cycles and networks of reactions, we would be able really to build like little microscale factory of parts, where we can really control what kind of shapes and properties of microscale assemblies we make. And so over the past years, I've been working on some of these problems with an overarching team of creating living matter in a jar from the bottom up, and basically a search for minimal ingredients that allow us to mimic the processes I described in the previous slides. And so in my lab, together with my collaborators, we work on several different things. So we work on trying to build artificial catalysts from the bottom up, trying to understand what are the necessary ingredients that we need to achieve efficiency of enzymes, so nature's catalyst. Then we are trying to understand what makes an artificial chemistry a good chemistry in terms of the number and types of building blocks, types of reactions that are there to find, for example, the minimal ones that are needed so that the system of coupled reactions can exhibit evolutionary adaptation. Next, we work on self-replicating structures, trying to understand what are the ingredients that we need to replicate any arbitrary structure. And finally, we work on polymer folding, and this is where I would say a few more words. Okay, so folding is connected to the process of self-assembly. So as I mentioned before, self-assembly is a process where pre-existing components, building blocks, spontaneously come together and form complex structures with high fidelity and high yield. So this is something that's very ubiquitous in nature. And so how do we explore this process in artificial systems? Well, we take a model system and we try to understand what happens, how things self-assemble in this model system. So the one that I work with, as I mentioned, is based on spherical particles. And then in experiments, if you just take a bunch of identical spherical particles and you put them in a solution, they will just aggregate together. And principally, if there are many, they will make a crystal. But if you limit the number that you put in, if you just take a few, they're going to make various kinds of compact structures. And so what I'm showing you here is what kind of structures you can get in 2 dimensions and 3 dimensions, well, 3 dimensions, 2 dimensions. And so the nice thing about these compact structures is that they can be completely enumerated up to certain size. And now, so going back to the nature self-assembly, the building blocks that come together have very specific interactions, okay? Because, for example, in this image here, these are ribosomal RNA, and then these red parts that are marked are places where specific proteins come and attach to make the ribosome. So if you want to make something very specific, we need to put in specific interactions in our system. And we can do that by coating the particles with DNA, as we discussed before. So to make these molecules, so we do the following. So for example, let's look at the system here where we have 8 particles. So there are 13 different compact structures that can be spontaneously assembled if all particles are identical. And now we want to make one specific one. We have to introduce specific interactions between particles. And so what I'm showing you here is one particular example of what we call chemistry, which tells you basically that if we take 8 types of building blocks that follow these interaction rules, where gray means that particles have complementary DNA strands, they like each other, and white means they don't. We take 8 particles following these rules and we put them in a solution, they will spontaneously assemble only this one structure that we have here. Okay, and so now there's been really a lot of theoretical and experimental work on questions like this, and I'm just listing here some of the names where people are trying to solve this so-called inverse problem. But the bottom line is that if we want to reliably assemble a unique product with high yield, we either need large sets of different building blocks or very complex building blocks in terms of shape and valence. And if you look to nature, this is not what nature does, right? So nature uses just 20 types of building blocks, basically amino acids, arranged, so chained in polymers and in a specific way, so following a specific sequence. And then these polymers fold into final 3D structures with high yield and fidelity. And so how would basically the self-assembly through folding look in artificial systems? And so we teamed up with the group of Jasna Bruic to work on this. So we are focusing here on 2-dimensional system at the moment. And as I said, we can enumerate all possible structures up to a certain size. So I'm just showing you here a zoom into how many structures you have at size 7. Now, what we can also make is what we call potential energy folding landscapes or folding trees. And that is, if we start from a chain of n particles, we can find what are all the structures that can be formed by sequentially adding bonds until specific compact structures are formed. Now, this is an example where we start from a polymer of length 7, and at each level, there are structures that have the same number of bonds, and the sequence of bond formations makes what we call a folding pathway. Okay, so now this is when everyone is identical. So now our next step is to try to add specific interactions and to see how we can actually build something high fidelity, but with not a lot of building blocks. Okay, and so we can control the number of different interactions, but we can also control when we turn on and off these interactions. And this is what I mentioned before, by designing the DNA sequences, we can select the temperature at which they are available. And so what I'm showing you here is that if we just take two types of building blocks and we impose certain protocol of how we turn on the interactions, the three available interactions for two different species, you can actually make the system fold into one specific structure and not the other ones. And so this is just a summary of what we've done so far. And so up to size of 13, there are over 600 different structures one can make. And then with only 3 colors and by varying sequence and protocols, we can reliably assemble over 300 of these structures, which is sort of really amazing, because if you actually start from like freely floating particles in a solution, and 13 of them, the only way to make something that's of size, very specific thing of size 13, is to take 13 different building blocks. And here we can actually do it with 3 and quite reliably. And so these were all theoretical predictions. But of course, we did this also in experiments and confirmed in simulations that they're calibrated to experiments. So I'm showing you just here 9 structures that can be formed if you start from sequences which have alternating flavors. And here they're just at the bottom are folding movies of both in simulations and experiments of a specific structure. Now, the questions that we are interested in are which geometries are more designable than others. We want to understand the folding pathways, how I can direct the system to make a specific structure, what is the kinetics of the process, and so on. So there are really lots of questions that we are exploring. And in the end, just a teaser, we basically, what is interesting that once we know which compact structures we can get through folding, we can start also exploring how one can make higher order structures like protein complexes. And it's interesting to see that if you just focus on having one type of this higher order building block, that the structures that we see forming are either something like these tubules and fibers, which we see in biology a lot, or these dimers made out of space invaders. And if you don't know what space invaders are, you are quite young. Okay, so I finish and I repeat this slide because it really summarizes what I hope I managed to convey in this short time, how we are exploring the unknown in research of these minimal ingredients and trying to find these laws and rules. And of course, these are my collaborators. The work that I've told you about has been done together with the group of Jasna Bruic and her postdoc, Angus McMullen, and with my PhD student, Maitana Munoz-Basaguit. And so I thank you for your attention, and I'm happy to take questions later on when we have the Q&A session. Thank you.